Hi, I'm Keith Ward. I'm the CEO of Kuria Therapeutics. We're developing novel medicines that power sight. We say that because we focus on the biology of the mitochondria. And our core therapeutic hypothesis is that mitochondrial dysfunction is a root cause of multiple blinding diseases. Of course, we're all taught in Biology 101 that the mitochondria are the powerhouse of the cell and produce energy in the form of ATP, which is correct. But what's less well understood often is that in the course of that production, waste products are also created, namely reactive oxygen species, or ROS. And the mitochondria must constantly balance production of energy with management of these waste products. The cells develop elaborate processes for detoxification of reactive oxygen species, but when there's ROS overload, whether that's an environmental insult, some underlying disease state like diabetes, or a chronic condition like of aging, the mitochondria are forced to wind down ATP production and therefore cellular function to be able to manage that ROS load. And it's this exquisite balance that the mitochondria constantly face in order to maintain appropriate function. Of course, that's no more important than in the eye. I think we all appreciate that the process of vision demands a lot of energy. In the retina, for example, ATP drives the visual cycle in the RPE. It drives photoreceptor turnover. It drives the neural retina signaling, but also in the cornea, which some people consider sort of a passive transparent window into the eye. But in fact, the only reason that the cornea is transparent is because of the very heavy load of ATP-driven processes that pump water out of the cornea all the time to keep the cornea transparent. And of course, the eye faces perhaps the largest load of reactive oxygen species in the entire body. In the retina, very high oxygen tension exists in the choriocapillaris. And in the cornea, literally every time we open our eyes, we expose ourselves to a high load of UV insult, UV being a potent oxidative stressor. So it wouldn't be surprising that the literature is replete with examples of the role of mitochondrial dysfunction in various ophthalmic diseases, whether that's in the back of the eye in glaucoma or age-related macular degeneration, or in corneal disease, including Fuchs dystrophy, where the relationship between mitochondrial dysfunction and that disease is direct and has been very carefully worked out. So we founded Kuria Therapeutics to focus on this biology and specifically on the role of a key protein called NRF2, which is a master regulator of mitochondrial function. And we develop novel activators of NRF2. We're not alone in this insight. In fact, there are over 17,000 papers elaborating the role of NRF2 in mitochondrial function in biology and over 500 in the eye alone. And the net of those is that activating NRF2 works on both sides of that balance. Activating NRF2 increases ATP synthesis, drives new mitochondrial production to drive more ATP, and in fact, powers sight in a literal way. But also, it's probably even more well-known function is as the master regulator of ROS detoxification. Activating NRF2 restores redox balance. It reduces NF-kappa P-driven inflammation. It reduces fibrosis, and it helps to clear mitochondrial waste products. So in a way, activating NRF2, as I said, works on both sides of that cellular balance. We believe we have best-in-class ophthalmic NRF2 activators, and our lead agent, called SCO-116, is an advanced molecule that's got potent, selective, active NRF2 in animal tissues, including in the eye. We've developed a preservative-free solution formulation that's ready to go to the clinic. We've got good composition of matter patent coverage. The agent is well tolerated, including on repeat dose ocular administration. We've got good efficacy data in preclinical models. We've shown that we can improve mitochondrial function, as you would expect, with an NRF2 activator, resolve fibrosis, and we have activity in gold standard models of ophthalmic disease, including dry AMD. And the agent is ready to move into GLP toxicology now and will be phase 2 ready directly within 12 months. Where we're going in the clinic is both retina and cornea focused. In the retina, we're interested in dry AMD, where the biology of mitochondrial function and oxidative stress has been elaborated for multiple decades. We believe we have an opportunity there to improve visual acuity and intervene early in the disease process. In the cornea, we're interested in 2 lead indications. One is Fuchs dystrophy, where there are also no approved therapeutics, and competitive programs primarily focus on end-stage corneal disease. We plan to intervene early in patients with Fuchs to improve their vision by powering their sight. And also, we have an interesting indication as an adjunct to cataract surgery to protect the corneal endothelium from the very heavy Ross load evoked by phacoemulsification. Of course, we have a multitude of indications that we could pursue and that we're interested in pursuing. We have multiple follow-on molecules in addition to SCO-116, and we intend to pursue all of these in time. Thanks for letting me introduce the company to you. We're Kuria is powering site. Happy to address any questions. Feel free to reach out. Thanks.